Okay, it's four minutes past the hour. I'm Iris Flores of STEM Biosis, and we are your host for today's session. This is part 2 of our 3-part series. And today we're going to cover culturing iPS-derived cardiomyocytes on cell-derived matrices like Salvo MatrixPlus. And I'd like to first start out by introducing Dr. Travis Block of STEM Biosis.
Hi, I'm Dr. Block, Chief Technology Officer at STEM Biosis. I've been with the company for just over 5 years, and prior to that, I was working at the company's core technology for the preceding 5 years. And I'm here today with my colleagues from CARTOX and the University of Michigan, Todd Heron and Andre Montero de Rocha. Dr. Heron is the director of the Frankel Cardiovascular Regeneration Core at the University of Michigan, the founder of CARTOX, which was recently acquired by STEM Biosis, and he's a paid consultant as well for STEM Biosis. Dr. de Rocha Montero de Rocha is faculty at the University of Michigan as well. He's been doing extensive work on the culture of iPSC-derived cardiomyocytes on our newest product, Salvo MatrixPlus. And he's also an expert in developmental biology and is also a paid consultant with STEM Biosis. And so welcome to the second part of our seminar series on the use of iPSC-derived cardiomyocytes for cardiac testing in vitro. And today we're going to be speaking about culturing iPSC-derived cardiomyocytes on extracellular matrix products. During this webinar, please feel free to type questions into the chat function. I'll do my best to answer as we go, but at the end of the webinar, I'll also read questions aloud for answers from either myself or Drs. Heron or Montero de Rocha. With that, I'll go ahead and get going. And so the kind of way this talk's going to work today is I'm going to start by talking generally about the importance of extracellular matrices. I'll dive into, and then we'll dive into the culture of iPSC-derived cardiomyocytes on MatrixPlus, and from there we'll discuss current protocols and recommended uses of MatrixPlus for human iPSC-derived cardiomyocytes. So I always like to begin my presentations. Yep. That one had a audio still. So I'd like to begin today with just a very quick history lesson on where cell culture comes from. And so these two people, if you're not familiar with them, you probably should be, because they're the founders of modern mammalian cell culture. So Alexis Carrel was actually a vascular surgeon who won a Nobel Prize for developing the techniques that led to the very first organ transplants. And Montrose Burroughs was a student in his lab. And they were studying capillary bed structure. And so they were taking thin sections of tissue and doing microscopy. And when doing that, they noticed that cells migrated out of the tissue and onto the slide, and that they were able to keep those cells alive by continuing to bathe them in nutrients. And so in 1910, they published that they tried to establish a general method to permit the culture like that of microbes, of tissues, of adult organs, of animals up to man. So bacterial cell cultures had already been in existence for hundreds of years, people making bread and beer. But this was the first instance of someone demonstrating that they could keep animal cells alive outside the body. And they actually published a paper describing the first immortal cell culture, though it turned out later that their methods were actually introducing new cells every time they did a media change. So that turned out not to be true. But I like to point out that while these two really stumbled across a finding that is the foundation for everything we're doing today in mammalian cell culture, they also, in a lot of ways, set us back, because they made this finding by accident, we were stuck with these processes that really make no sense. And still today, our processes look a lot like what they developed, where we tend to dissect a tissue, and we tend to then disrupt that tissue either enzymatically or physically to get at cells of interest. And then we stick those cells onto very simple substrates. And often this is tissue culture plastic, or perhaps it's a thin coating of fibronectin or collagen or laminin, or a mixture of a few extracellular matrix proteins. But whatever the case may be, we're taking cells out of their native environment and putting them onto a tissue culture dish and just kind of relying on the fact that they stick to it. But cells react to their microenvironment. And so these cells change phenotype as soon as we put them into these bizarre culture conditions. And this is really a product of this accidental discovery more than 100 years ago. And so what I really want you to take away from that story is that just because we've been doing something, in this case, cell culture, one way for a long time, we shouldn't assume that it's the only and definitely not the best way to do this. And I think this is really important to mention because, you know, we're taught from a very early age in biology, and it goes on really all the way through college and graduate school and in research that cells are really the basic building block. But in the body, they're constantly interacting with extracellular matrix. And actually, cells make up just a tiny part of the volume of your body, and you're really mostly extracellular matrix. And so this is an SEM image of a cell in its native environment. And you can see all the communication that's going on across this cell membrane. And you can actually see, well, when they did the freeze fracture, you can actually see these pores where extracellular matrix had been interacting with the matrix and it got ripped out. And then when you take a cross section of this, until you label it, it's actually really difficult to tell where the cell ends and the extracellular matrix begins. And that's because the boundary really is very blurred, because extracellular matrix components are interacting directly with cytoskeletal components to impact cell function, cell fate. And so in this image, going from left to right across the screen, we actually have a phospholipid bilayer going diagonally across. And on the left, the bottom left, we have the cell, and on the top right, we have the ECM. But despite this vast amount of communication going on between cells and their extracellular matrices, we still have these extremely simplistic cell culture systems. But then in the last 2 decades, really, we've, as a field, have begun to pay more attention to the role of microenvironment in regulating cell fate. And so people have started to really pay attention to how these different extracellular cues impact cell function. However, even as we've paid increasing attention to this, the models that we use to study the microenvironment and to optimize microenvironmental factors for specific applications of cell culture tend to be extremely simplistic. Oftentimes we're manipulating a single variable at a time. For instance, somebody might make an acrylamide gel and coat it in fibronectin so that cells can stick to it, so that they can use the ratio of acrylamide and disacrylamide to manipulate the stiffness and determine what the optimal stiffness is for a given application. However, my favorite study that I put on this page was actually this one in the very bottom that was published in PNAS in 2010. And the reason it's my favorite is it shows that these simple models really fail to understand how there's a dynamic interplay between these factors. And so in this study, they actually made little cookie cutter cutouts in their cell culture dishes, and they put MSCs into that dish, and the cells were then forced by the topography to assume the shape of those cookie cutter cutouts. And by changing the shape, you could actually change the gene expression in the cells and force them into an adipogenic or an osteogenic lineage. And so with all the same biochemistry, with the exact same mechanical properties, structure changed how cells responded to those to that biochemistry and those mechanics. And so we can't expect to isolate these variables and be able to come up with the optimal environment, because there's a dynamic interplay between all of these properties. And cells are changing how they respond to stiffness based on what cytokines are present, based on the density of cells, based on the structure, and based on the biochemistry of the matrix and where binding sites are and what exactly those binding sites are. And with that, I'm going to transition into a brief overview of our approach to addressing this really dramatic shortcoming of traditional cell culture approaches. So for starters, I want to kind of define the goal. So an ideal extracellular matrix, or ECM, should be able to elicit a biologically relevant cell function. So in standard culture conditions, really rarely elicit biologically relevant cell phenotypes. When we're putting primary cells onto them, we often talk about de-differentiation of those cell types. And when we're talking about stem cell derived cell types, frequently we're able to differentiate those cells into fetal-like cells, but they rarely, if ever, mature into biologically relevant cells that resemble cells found in an adult human. And so an ideal ECM for in vitro applications should elicit biologically relevant cell function. It should be tissue specific because multiple microenvironments exist in the body. And so a one-size-fits-all solution tells us very little about the function of those niches. And if it's eliciting a biologically relevant phenotype, it may be that we're pushing it too hard down a certain path with the components in there. And so we want it to be tissue specific so we can really truly elicit biologically relevant phenotypes. And it should have physical and biochemical properties that mimic in vivo conditions. And then lastly, we should be able to not just recreate healthy phenotypes, but we should be able to recreate diseased phenotypes so that we can actually do effective drug or toxicity testing in vitro. So our approach that we use for our core technology at STEM Biosis is that we take matrix producing cells, we seed them onto tissue culture dishes, we allow those cells to grow to confluence and secrete a matrix, and then we use a low concentration non-ionizing detergent to remove those cells, leaving behind the matrix that they secreted. And so the matrix they secreted is actually visible in bright field. And so you can see here, this is nothing like doing a fibronectin coating or a matrigel coating. You can actually see the fibrous structure that was laid down by the cells in the dish, but the presence of that fibrous structure does not interfere with our ability to image cells on that matrix in bright field, nor does it interfere with our ability to do fluorescent imaging. So we have a natural environment that was secreted by cells, and it fits into existing workflows. So when we ship our Salvo matrix to customers, we're shipping it actually, it looks like tissue culture plates. However, the bottom, the culture surface of those plates has a thin coating of cell-derived matrix that just is air dried on, and it has a 2-year shelf life. So all the end user has to do is add a little bit of PBS to rehydrate the matrix, wash that away and add their cells. And so it's very simple to use. And then what's most important is that we've seen that for a variety of cell types, we're able to elicit more native phenotype from cells cultured on our matrices. And so we're able to get biologically relevant results in vitro that can better translate to in vivo conditions. And importantly, we've been able to validate this model in multiple systems in peer-reviewed papers. And so we've now done this for bone, fat, cartilage, pancreatic cells, and heart cells that have all been published on by our group and by related groups. And so just going a little bit into how we validated this in different systems. In the left panel here, we have a very simple OB and osteogenic and adipogenic differentiation assay. And here we showed that a bone marrow derived extracellular matrix supported osteogenesis, whereas the adipose derived matrix inhibited osteogenesis and promoted adipogenesis. And so cells on those matrices, it was driving their fate decisions, and it was driving it in a predictable manner based on the lineage of the cells that were used to produce the matrix. On the right-hand panel, we produced an articular cartilage-derived extracellular matrix. I should say articular chondrocyte-derived extracellular matrix and bone marrow MSC-derived matrix. And we showed that the articular chondrocyte-derived extracellular matrix could better support phenotype retention of primary articular chondrocytes, such that they expressed high levels of collagen II and glycosaminoglycans after passaging on our matrices. And that's in contrast to traditional culture methods where there was essentially no collagen II expression. some of our most recent findings that Todd and Andre are going to go more into today are that our newest product, Salvo MatrixPlus, can be used for the rapid and spontaneous maturation of iPSC-derived cardiomyocytes. On the left-hand side, we have an image of iPSC-derived cardiomyocytes on Matrigel. This is what's typically seen in cell cultures today, where there's really no directionality or organization to it, and the cells have a fetal-like phenotype. Whereas on the right, after 7 days in culture, the cells have aligned, formed a functional syncytium, and are rod-shaped, as you'd expect to see from mature cardiomyocytes. But again, Todd and Andre will comment more on that in their portions of the talk. And so I hope at this point you appreciate that these matrices are capable of eliciting more biologically relevant phenotypes. And so now I just want to briefly touch on the fact that we're actually able to produce tissue-specific matrices. And so this data right here comes from a 2016 paper from Dr. Marinkovich at the UT Health Science Center in Dr. Chin's lab. And it's take bone marrow and adipose-derived MSCs and produce matrices from each of those. And we showed that just from MSCs from different sources that are frequently talked about as being interchangeable cells for a lot of applications, the bone marrow derived MSCs actually produced a matrix that was about 2 orders of magnitude stiffer than the adipose MSC derived matrix. Not only that, it was more tightly aligned, whereas the adipose matrix was much more isotropic. And there were some key functional differences or biochemical differences between the matrices. However, what's perhaps most interesting about this is not the differences, but the similarities. And so these matrices actually shared more than 80% of their composition, yet they had dramatically different structure and mechanical properties. And that manifested itself in different functions, which I showed earlier. And so now I'm going to talk a little bit more about MatrixPlus, which again is the matrix we're using for cardiomyocyte maturation, and that's what Todd and I are going to present data on today. And so this is a matrix that we began producing about 4 years ago for a collaboration between our labs. And we see here that MatrixPlus has a distinct structure that you can visualize via the topography, as well as this bright field image from our original Salvo Matrix product. It is also significantly more adhesive, and this is a measure of surface energy, and it is much, much less stiff. And so this matrix is on the order of 5 kilopascals in stiffness, much more similar to our adipose-derived matrix. And then I'm not going further into these differences in the mechanical, structural, and biochemical properties, because it's very difficult to tease out a mechanism of action here. So one reason that I think matrices often get ignored in cell culture applications is that they're very difficult to study, because as soon as you manipulate one property, you're likely changing more than that. You know, you can't take out a major component without expecting to change the structure and mechanics of the matrix. And so because of this, we believe that this biomimetic approach is the best way to create biologically relevant cell culture environments. Here, we show that there's some significant differences in the biochemistry. And again, it's very difficult to tease out a mechanism of action, but we do know that using hierarchical clustering, there's consistency in the differences between the two matrices, and that Salvo matrix is collagen rich and proteoglycan poor relative to MatrixPlus. One potentially key difference that we're interested in studying in the future is that MatrixPlus is very rich in perlecan, or also known as heparin sulfate proteoglycan 2. And one reason why this is interesting in the context of cardiomyocyte maturation is that mutations in that gene, in the gene that encodes perlecan, lead to early death, often caused by cardiac malformations. So that's an interesting piece we were able to tease out of this data that is, potentially an interesting hypothesis for a mechanism. And so before we move on to Todd's portion of the talk, I just want to give a brief overview of Salvo matrices for cell culture. So cell culture substrates are absolutely critical, but they're often misunderstood or totally ignored in their role in cell culture. And then in vivo, ECM plays a critical role in determining cell state and function. And then in vitro, nuanced differences in ECM properties really almost nothing for the cardiomyocytes, whereas MatrixPlus has a profound, though about 70% of the composition is shared between the two. And then in vitro, cells may be induced to secrete a matrix that mimics the structure and composition of their tissue of origin. And then lastly, tissue-specific extracellular matrices are a robust model for listing biologically relevant phenotypes in vitro. And maturation of iPSC-derived cardiomyocytes on Salvo MatrixPlus. Todd, you're on mute.
Hello, I'm happy to be here today to present some work that my lab has done over the years using the Salvo MatrixPlus extracellular matrix from STEM Biosis. So the results that I'm going to be showing have been published. The title of that publication is here in the bottom left. And it's Human Perinatal Stem Cell Derived ECM enables rapid maturation of human stem cell derived myocyte structural and functional phenotypes. And so what we did when I first started working with STEM Biosis, I was realizing in my own lab the importance of the extracellular matrix for cardiomyocyte maturation. And so I agreed to do a study that you see here. And the experimental design was to use commercially available cardiomyocytes from Cellular Dynamics, which arrive in the lab cryopreserved. And then what we did to test the effect of different matrices was we plated those cells as monolayers in 6-well dishes, where each monolayer contained 200,000 cells per well. We used 3 different matrices. First, we used a matrix that is commonly used in the field and used widely in my laboratory, and that's Matrigel. That's a mouse-derived extracellular matrix. We also used a product from STEM Biosis called Salvo Matrix, which is a human-derived matrix. And then third, we used MatrixPlus, which is a newer product from STEM Biosis, and this matrix is derived from a perinatal stem cell source. In the results that I'll show you, we did some basic microscopy experiments looking at the cardiomyocyte structure, and we also did some electrophysiology and medication screening to look at the effect of the ECM on the cardiomyocyte function. So this is a phase contrast image of a monolayer of Cellular Dynamics cardiomyocytes plated as a monolayer on Matrigel. And here you can see that the cells are round in their morphology and have no real direction to them within the cells or between the cells. And this indicates an immature fetal-like cardiac phenotype. When we used another matrix from STEM Biosis, their Salvo matrix, this matrix is produced from bone cells. We saw similar effects where the cardiomyocytes cultured on this matrix are also fetal in structure. But when we looked at the same batch of cardiomyocytes that were plated on the Biosis MatrixPlus products, we saw a much different morphology of the cardiomyocytes. We saw a much more rod-shaped morphology of the cardiomyocytes, and we also could start to observe some alignment of the cells from cell to cell within the monolayer. So this triggered us to explore in greater depth and greater detail the effects of these matrices on the function and the structure of cardiomyocytes. And so here you see a nice phase contrast image comparing the same batch of cardiomyocytes cultured on Matrigel on the left compared to the same cells cultured on MatrixPlus on the right. You can see very dramatic differences in the shape and the alignment of the cells induced by the ECM of the MatrixPlus. We were really excited and enthusiastic about continuing to use MatrixPlus because when we looked at higher magnification, we could actually start to see the light-dark repeating pattern of the cardiac sarcomeres, which is typical of adult cardiomyocytes, but not typical of fetal immature cardiomyocytes. So what we found was really exciting, that these cells can mature on MatrixPlus just 7 days after being thawed. The MatrixPlus coating is compatible with standard microscopy, as Travis alluded to, and you can see in the image here. And another really exciting aspect of using MatrixPlus is that the plates that you're going to use are already coated and have a very long shelf life. So here this shows more of a bird's eye field of view of the cardiac monolayers. And again, here in these pictures, you can see that the cardiomyocytes cultured on Matrigel have circular shape with no directionality, but the cells cultured on MatrixPlus in the monolayer, they start to form fiber alignments where the cells are actually lined up relative to one another in one direction. This is shows the result of immunofluorescent staining and confocal imaging to look at the structure of the sarcomeres of the cardiomyocytes cultured either on Matrigel on the left or the corresponding stain for cells cultured on MatrixPlus on the right. And so for 3 different sarcomere proteins in A, troponin I, in panel B, cardiac troponin T staining, and in panel C for alpha-actinin staining, you can see that the Matrigel cells are radial in structure or circular in structure, whereas the same batch of cells but cultured on MatrixPlus take on a rod-shaped morphology, where the sarcomeres are aligned and parallel to each other throughout the cell. And also interestingly, the majority of cardiomyocytes on MatrixPlus become binucleated. And this binucleation is characteristic of cardiomyocytes in the adult heart. And here in panel D is a quantification of the circularity index, showing that the cardiomyocytes cultured on Matrigel have a high circularity index, as the pictures would indicate, and that the cardiomyocytes cultured on MatrixPlus have a much lower circularity index. Here, we did some more structural analysis, looking at the cytoskeletal organization and also at cardiac troponin I expression in the monolayers. And so in these multi-panel figures, what you can see, if you focus on the phalloidin staining in green in panel C for Matrigel compared to panel G for the cells on MatrixPlus, you can see a much more structured and more ordered cytoskeletal organization from cell to cell when the cells are matured on MatrixPlus. And then if you focus your attention to the red channel in each panel, panel B for Matrigel and comparing to panel F for MatrixPlus, this is staining for a protein called cardiac troponin I. This is a component of the mature myocyte myofilament, And also cardiac troponin I is used as a biomarker for any kind of damage or cardiotoxicity. And you can see that the cells cultured on MatrixPlus express much more cardiac troponin I and much more ubiquitously throughout all of the cells in the field of view. And this is important, especially for any sort of biomarker analysis that might be done in vitro to try to detect CTNI as a biomarker of damage. So we also went on to examine the mitochondrial distribution and function in monolayers of human cardiomyocytes cultured either on Matrigel or Salvo matrix. And what we find, and what you can see in the fluorescent images, is that MatrixPlus promotes mitochondrial distribution throughout the cardiomyocytes. This is easiest to see in panels C and D. So the cardiomyocytes here stained with mitotracker red to label the mitochondria. You can see that the immature cells cultured on Matrigel, the mitochondrial signal is huddled mostly around the nucleus, with very little distribution throughout the cytosol of the cardiomyocytes. However, the cells on MatrixPlus have much more robust staining for mitochondria and much more mitochondrial distribution throughout the cardiomyocytes. And in fact, when we do a comparison to adult mouse ventricular myocytes, we can see that the signal and the quantification of the mitotracker signal is starting to approach the values of the adult cardiomyocytes. So next we went on to do some electrophysiological analysis using cardiac optical mapping to detect drug-induced arrhythmias in vitro. So the movie that you'll see here on the left of your screen is the baseline recording of action potential propagation through a monolayer. This is a large monolayer, about 1 centimeter in diameter. And you can see very nice propagation of the wave uniformly throughout the monolayer as the wave spreads from left to right. When we add to the same monolayer hERG channel blocker, this is a compound or a drug that causes arrhythmias by prolonging the action potential duration, we saw a much different activation pattern, where the activation pattern became a very stable rotor, spiral rotor or spiral wave that is reminiscent of reentrant activation pattern that would occur during fibrillation of the heart. This is exciting because it actually shows that we can record and observe fatal torsade de pointe-like arrhythmias in vitro, where it's induced by a medication. The plot in the center shows the time course of the effects of E4031 to cause arrhythmias. So here you can see the application of E4031 after the first beat. And then after about 4 beats, we could start to detect action potential duration prolongation, which progressively increased and also coincided with the occurrence of early after depolarizations, which you see here labeled in red. And then finally, what you see in the movie as the stable rotor is a TDP-like arrhythmia, which was maintained very stably. So this shows and summarizes that the effect of the hERG channel blocker to cause torsade de pointe-like arrhythmias was only observed when cells were grown on MatrixPlus. So the red traces, these are action potential measurements of monolayers cultured on MatrixPlus on the left at baseline, and then in the presence of E4031, the potent hERG channel blocker. For the Salvo matrix, which is a bone cell derived matrix, you can see that the E4031 did have an effect, but that effect was prolongation of the action potential without any TDP-like or rotor formation or occurrence. And in the case of Matrigel, you can see again that the effect of E4031 was to prolong the action potential duration, combined with EAD formation. And in the case of Matrigel, we did see TDP occurrence in 1 out of 6 monolayers. So MatrixPlus creates a substrate where one can induce torsade de pointe like arrhythmias using medications that are known to have that effect in vivo. And then just to make sure that the assay that we were developing on MatrixPlus wasn't too sensitive of an assay, we used another hERG channel blocker called ranolazine. This medication is a hERG channel blocker, but is considered a safe medication because it also has effects on other ion channels and doesn't cause arrhythmias in patients. And so you can see in the case of MatrixPlus that when we add ranolazine at a high dose, we don't see any arrhythmias, but we do see a slight prolongation of the action potential duration, as you would expect for a drug like ranolazine. We went on to also do some microelectrode recordings to quantify the membrane potential changes during the activations and the spontaneous activations of the monolayers. So here is a summary of some of the more important features for maturation of stem cell-derived cardiomyocytes. One is the maximal diastolic potential, and it's hyperpolarized compared to historical data. It's minus 79 millivolts, which is very similar to that of cardiomyocytes from an adult heart. If we look at the action potential upstroke, or the change in voltage with time in the upstroke of the action potential, which is caused mostly or attributed predominantly to the sodium channel function, we can see a very rapid upstroke of 234 volts per second, which is very similar to the values obtained in adult cardiomyocytes. The APD90 is shorter and similar to human cardiomyocyte action potentials from native hearts. And finally, when we look at the conduction velocity of the action potential through the monolayers, we can also see rapid conduction velocity of around 35 centimeters per second. At STEM Biosis, after we found these results that were very promising for maturation of stem cell-derived myocytes, at STEM Biosis, they began to generate 96 well plates coated with MatrixPlus. And we did that so that we could do higher throughput screening and do a more careful validation of medication effects of the cardiomyocytes cultured on MatrixPlus. So routinely in the lab now, we perform 96 well plate optical mapping. An example of that is shown here, where each well is loaded with a fluorescent dye to measure the action potential. And each monolayer has its own pacemaker, and we can record from all 96 wells and create very robust data sets with very high numbers and very good resolution. So to validate the 96 well plates, we started by looking at the average baseline beat rate of the cardiomyocytes over 7 different multi-well plates, which you can see in panel A. So the average beat frequency was around 1 hertz or 60 beats per minute, which is a very good resting beat rate for these cells. And you can see also that there was very little variability or variance in beat rate from plate to plate, with the average variance from plate to plate averaging just about 3% variance. So it's very reproducible and very robust data that can be collected. So we also started to validate this assay and the cells on MatrixPlus using SIPA validated compounds. So these are compounds that are medications that have clinical data to support their risk for either causing arrhythmias at a high risk, an intermediate risk, or low risk. And here we compared the total incidence of arrhythmias for 12 different compounds, about 4 from each group. And in the low risk, we can see no arrhythmia occurrence. With the intermediate risk, we can see the red starts to creep up, and we see more red with MatrixPlus compared to Matrigel, suggesting a more sensitive assay using MatrixPlus. And then if we look at the culmination of results for high-risk compounds, we can see that at these doses, which is 10 times the effective therapeutic plasma concentration of these drugs from clinical data, that we can detect many more arrhythmias when the cells are matured on MatrixPlus compared to the cells cultured on Matrigel. So here, this shows a more detailed analysis of the SIPA compounds that have been used and validated using MatrixPlus. Here we show the number of wells out of 6 wells at a dose of 10x the effective therapeutic plasma concentration. And you can see that when we plot the number of arrhythmias, when we use MatrixPlus, where the arrhythmias are shown in red, compared to the same cells cultured on Matrigel, where we see much fewer arrhythmias when the cells are immature on Matrigel. So this suggests that the MatrixPlus provides an ECM where the cardiomyocytes mature and provide a more sensitive proarrhythmia test and cardiotoxicity test. So for a summary, we've shown, and we're very pleased to find and to use MatrixPlus because it offers a human ECM with properties that mature cardiomyocytes in just 7 days. We've shown that MatrixPlus promotes mitochondrial maturation, functional maturation, and also sarcomeric maturation in 2-dimensional monolayer culture system. And then I also showed data from optical mapping. And in the next webinar in 2 weeks, we'll be discussing more about the CARTOX imaging system and the use of this system for high throughput screening using cardiac action potentials and calcium flux as a functional readout. And so that's the conclusion of my portion. I'll hand it over now to my colleague, Andre Montero de Rocha, who will talk about the uses of MatrixPlus.
Hey, Andre, please make sure you're unmuted.
Hello, can everyone see my screen?
Yes.
Hello?
Yes.
And you can hear me too.
Yes.
All right. Yep. Thank you, Todd and Travis. So at this part of this seminar series, I'm going to talk about current protocols and recommended use and applications of MatrixPlus. As Todd mentioned, it is a pretty new product, but we already know how it works in many different platforms, and also what are like really interesting applications that we can use plates that were coated with MatrixPlus. So if you're interested in using this this product, it is pretty simple to use, and somehow my slides are not moving. Let me try this. Okay, so I hope that not like 5 slides move on. But anyways, so if you're interested in using MatrixPlus for culturing your cardiomyocytes, I'm actually like really happy to say that you don't need to have like a huge expertise in cell culture, and you don't need to juggle cells or anything. It's like really simple. And there are 2 recommended protocols that you can find online. One is provided by Biosis, and it's based on the protocol that we use here in Michigan. And the other one is a protocol provided by Fujifilm CDI that they developed for their cells, their product. And I'm gonna just now talk quickly about some general guidelines for when you're plating these cells, and then I'm gonna compare the other two protocols, and we're gonna move on into some applications. So as a general guideline, it is important to note that all MatrixPlus plates, they are pre-coated plates that can be shipped at room temperature, which shows that these matrices are very stable, and the shelf life of them is 18 months. So if you're worried about getting a case of plates and saying like, well, I'm just gonna use one, well, these plates are gonna last for a while in your refrigerator. So as a general guideline, when you're trying to when you're gonna plate your cells on MatrixPlus, the first thing you need to do is to bring the plates to room temperature. And after that, you're gonna rehydrate your plates with your preferred buffer. Here we use HPSS, but we know that if you use PBS, it works the same way. After that, you will wash 2 times these plates after you rehydrate them. Then you need to get your thawed or purified IPSCMs, adjust the concentration, and in plating medium. Then you aspirate the HPSS and replace it with your cell suspension in your plating medium. Then you need to wait for these cells to attach, and attachment period depends on protocol. And after that initial period on plating medium, you can aspirate your plating medium and add maintenance medium, and you should culture the cells for at least 7 days after plating. It does not mean that these cells are not going to be ready for your application before that. This is what we recommend, and I'm going to talk about that at my last slide. So the protocol that was developed and is provided by STEM Biosis, and it's based on what we do here in Michigan, it's very similar to like those general guidelines. So we're going to bring the plates to room temperature, rehydrate the plates with HPSS, then wash them 2 times with HPSS. And here we use either CDI cells, especially for drug testing, because they are FDA approved and like reliable, and you already know how important is to have this kind of resource for drug testing, right? Or we also do a lot of in-house cells. So if you produce your own cardiomyocytes, you can also use MatrixPlus, and you're gonna achieve similar results, right? So then you have to adjust the volume of the plating medium to obtain the desired concentration, and you want to plate 230,000 cells per square centimeter. Just use this number to calculate how many cells you will need per well. Just to give you a rough estimate, this cell density determines that you should use 75,000 cells per well of a 96 well plate. It is important to note that most of the time, if you're using Matrigel, you only do 50,000 cells per well, but the cells tend to spread more and they're more round. And cells that are cultured on MatrixPlus, they are rod shaped, so you need more cells to cover the same amount of area. And our plating medium is very simple. It's what people call EB20. It's the embryoid body 20% of FBS, so it's DMMF12 with 20% of FBS, 0.1% of glutamine, 0.2% of non-essential amino acids. And here we add 25 micromolar of blebstatin, and we use that because it increases plating efficiency of the cells. And of course, it's optional. You don't need to add ROCK inhibitor, but if you add 1 micromolar of ROCK inhibitor, it tends to increase the survival of the cells and give you a better coverage on your monolayers. After we plate the cells, we keep them in culture for 48 hours before we remove and we wash off the EB20 with HBSS. And we do that because EB20 has blebstatin and you want the cells to start contracting. So you just wash to remove any trace of blebstatin that will delay the contractility of your cells, right? And when we use our culture medium here in Michigan and what is in the protocol provided by STEM Biosis, we use RPMI supplemented with complete B27. And we change media every other day, and the cells are cultured for 7 days. The protocol that is recommended by Fujifilm Cellular Dynamics is very similar to ours, but they rehydrate the plates in HBSS for 1 hour at 37 degrees in the incubator, so at 5% CO2. They also wash the plates for 2 times, and then when you're thawing your CDI cardiomyocytes, please follow their recommendations. Then our kit, when you buy the cells, it's a kit, so you're going to receive also the plating medium. So use the plating medium as they recommend, and they also recommend the same cell density, that is 230,000 cells per square centimeter. There is a important difference here is that their plating medium in their protocol, they say that you can keep the cells in plating medium for between 4 and 24 hours before you aspirate off and wash the plating medium and replace with the maintenance medium that they provide. Yesterday I was in a seminar and Ravi said that they have some other new products for maintenance of the cardiomyocytes. There are media for different assays that can be used as well. And they also recommend the culture of the cells for at least 7 days. So as a summary of what is available as protocols, the main difference between these 2 protocols is that the plating medium is different and ours contain blebstatin. The first media change for us is always 48 hours after plating, and with CDI's protocol, it can be between 4 and 24 hours. And the maintenance medium, that is RPMI with B27 plus, and for the CDI protocol is their cardiomyocyte maintenance medium. The minimal recommended period in culture is 7 days. But we know from cells generated here that you can keep the cells for up to 30 days. And I'm going to talk about that in a little bit. So there are some recommended use, and I'm making a differentiation between recommended use and applications. I'm going to touch on applications in a little bit. So as recommended use, you can use actually MatrixPlus. It's not the scope of this talk today, but you can use MatrixPlus for stem cell maintenance as well. The cells retain pluripotency and can be cultured for long-term on MatrixPlus. We know that some clients of STEM Biosis are starting to differentiate the cardiomyocytes on MatrixPlus as well, so it supports cardiomyocyte differentiation. But what I want to talk to you is a little bit about like rapid maturation of the cardiomyocytes and also long-term culture of the cardiomyocytes on MatrixPlus. As we all know that there are several methods and several different approaches to mature iPS-derived cardiomyocytes. You can do prolonged culture, like for 3, 6, 9 months. You can add different hormones to your cell culture medium. Some formulations, they change, they alter the energy source. So they don't have glucose, but they have galactose or some other carbohydrates or more free fatty acids in their composition. And as we do with MatrixPlus, there is like, you can alter the cellular interactions and also the the cell interaction with the ECMs to promote maturation. And there are some other changes that you can do, that like biophysical stimulation. You can either stretch your cells or pace them for 30 days, or you can change the substrate stiffness, as Todd was mentioning that his lab already had interested in the interactions with the cardiomyocytes with the extracellular matrix, and it started basically on changing the stiffness of the substrate to see if we could induce maturation of the cells. And of course, there are like 3D culture methods that are supposed to change the morphology and the electrophysiology of the cells. There are some drawbacks to some of those different approaches, because some of them are like very time consuming. Sometimes people do 30 days of electrical stimulation to induce maturation, or like the 3D culture, they can be labor intensive, or they are inducive of just a partial maturation, like in our own paper where we use a PDMS to change the stiffness of the substrate. We could get some change in the electrophysiology. We induced a partial maturation of the cells, but they still didn't have the right morphology. And a lot of them are very low throughput. As Todd mentioned, like now we do our assays in a 9.6 well plate, and I know that Biosis is able to do 384 plates as well. So what MatrixPlus offers is a fast maturation method, and in which we have electrophysiology and morphology maturation in 7 days, and that allows high throughput. And we can also mature either ventricular or atrial specific cells. Here in Michigan, we've been using protocols to have either ventricular or atrial cells, and we've been putting those cells on MatrixPlus, and we observe the same effects on maturation of the cells. And it also enables the use of mature cardiomyocytes on multiple applications. I want to talk a little bit about long-term culture of iPSCMs. So in general, people that are culturing these cardiomyocytes, they do that in a 7-day period, and then when it comes for the cardiotoxicity test, in general, you expose your cells for 30 minutes or 1 hour to a compound, and then you measure your outcomes. In this case, we were testing like for how long we could culture the cardiomyocytes on MatrixPlus, because this enables chronic studies using mature cells. So in this flow cytometry graphs here, I've used max purified atrial cardiomyocytes. So these are magnetic beads assisted cell sorting cardiomyocytes that we regularly use here. I used the SPS plating protocol. The cells were cultured on RPMI with P27, and we kept them for 30 days. And we had a purity of 94.7 in one of the samples and 90% on other of the in another sample that we ran. As I mentioned, we can also do the long culture with ventricular cardiomyocytes. So these are monolayers containing ventricular cardiomyocytes. These are phase contrast image in which you can see that the monolayers are preserved after 30 days, and that you can also get these cells that were cultured for a long term and use in different applications like calcium optical mapping with CalBrite, or use FlowVolt for voltage mapping of these cells. What I want to share with you is like some very interesting results, because we did those optical mappings, and this is voltage. And what we see is that we have, after 30 days, a preserved chronotropic effect of isoproterenol. So this is the baseline, and this is after 500 nanomolar of isoproterenol. And there is an increase in beat rate, which is expected. One thing that is very interesting is that when we do the calcium mapping, this is the baseline. So we have here the fluorescence intensity of the assay, and each peak represents intracellular calcium release. So after we measured those, we treated the cells with 500 nanomolar of isoproterenol and recorded the calcium transients again within 5 minutes. So we could see that there was an increase in frequency of intracellular calcium release. But if you also look at the amount of fluorescence after adding isoproterenol, we have a ionotropic effect in which you have a higher fluorescence that is an indication of higher calcium amplitudes being released into the cytoplasm. These are the quantifications of the amplitude of the signal. So this is pre and post isoproterenol, and you can see that there is over a 2-time increase in in the calcium intracellular release. And these are the paired comparisons, like each sample where they were in terms of calcium transient amplitudes before and after adding isoproterenol. And this is important because this is the expected effect of isoproterenol, and it's not, it's still controversial if the stem cells have this kind of behavior or not, because not everyone is able to see that kind of isotropic effect. So after telling you a little bit about the long-term culture, and again, I mean, we wanted to see for how long we could culture them. It doesn't mean that you need to culture your cells for 30 days, okay? It's just that it is possible to keep the cells alive and well and with high purity for 30 days. So I want to talk a little bit about the applications of MatrixPlus. And here for us, 2 applications that are very important are the action potential investigation, right? And here in Michigan, we do that with voltage sensitive dyes, mainly fluovolt. But you can also use other different dyes. And you can also do the investigation with MEAs using MatrixPlus. We also do a lot of intracellular calcium release assessment using calcium sensitive dyes, especially CalBright. We know that the cells that are on MatrixPlus, they can be used for current measurements with patch clamp. We have some colleagues that use that, and even in our first paper, it wasn't for specifically current, but as Todd mentioned, we did microelectrode recordings, and some of the patch clampers that I talked to, they say that actually the cells, they are rod shaped, so they are taller, they have more volume, and they're easier to patch because of that. These cells can be used for transcriptomic and proteomic assessment or analysis. And even for, if you're not doing like a high throughput proteomic analysis, you can still collect your protein samples for Western blots. And as Todd mentioned, the matrix is compatible with many different optical assays. So you can do a lot of image-based assays. So you can use fixed cells for immunofluorescence or immunocytochemistry. And you can even do live assays like apoptosis or measure mitochondrial potential with JC1. And one that will probably become very, very popular is to use movies to obtain motion and measure motion tracking. So you can have an estimate of contractility in 2D. So here, So after talking about all those applications, and those are just some of them, if you have something that is like very specific, like if you want to do, because I forgot to put here, if you want to do respirometry assays, there are seahorse plates that you can use, and there are many other applications that can be performed using MatrixPlus. So this is just to transition to what is available in terms of formats. So there are optically transparent plates that have thin bottom that will allow you to do imaging in confocal microscopy, for example, or use plate readers if you're using, for example, the Lycor Odyssey M system that does in-cell Western blots. That's how they call them. You can also use MatrixPlus coated MEA. So here you can see an image of an Axion MEA plate and the cells covering the electrodes. You can actually see that the cells still have their rod-shaped morphology. And this is an example of what I told you before. So this is an image that is a movie that was recorded of atrial cardiomyocytes that were on MatrixPlus coated cover slip. And you can use this to track motion and estimate contractility of your cells. So the plate formats that are available for all these applications are regular plate formats. So these are regular plastic with the thick bottom plastic plates. And off the shelf, they have 6-well plates, 9 6-well plates, T75s, and T150s. They also make off the shelf some special plates, like the ultra flat cover slip bottom plate. That is the one that has the square wells that I showed to you a little bit before. They also have other types of microscopy imaging plates that are also thin and confocal compatible. And they also have cover slips that you can put into a regular plastic cultureware dish and seed your cells on top of the cover slip. Actually, a lot of the immunofluorescence for the paper that Todd published, we still didn't have the high throughput formats, and we did those on cover slips. And actually, the other thing is that the patch clampers love the cover slips because they can put their cells on, they cut the cover slips, and they can do their patch clamping on fragments of cover slips, right? So there's also the possibility of customization of plates. As I've mentioned, we know that STEM Biosis has coated seahorse plates for people that want to use cells on seahorse plates that are with mature cells, and also MEA plates. If you're not really interested in any of those formats, or if you have an assay that is very specific, you can contact Iris Flores at stembiosis.com, and she will arrange with you how to attend your needs and coat your plates with MatrixPlus for you to develop your assay, or just adapt your assay to mature cells. There are three frequently asked questions that I'm already gonna put them out there because we actually really don't know the answer yet. One of them that we hear all the time is that, can I use the cells before 7 days? And you can, but we don't know if the cells are gonna have their full potential developed before that time. The other one is, if the cardiomyocytes are going to retain a mature phenotype, if you culture the cells on MatrixPlus, lift and replate them. So a lot of people that ask about that is because they want to culture the cells in a regular plastic cultureware and then put them into some other plate to be able to do imaging. So if your final outcome is a confocal imaging or something that needs a optical plate, you can buy the optical plates directly or buy the cover slips and use them instead of lifting and replating them. But this is definitely one thing that we're looking into, and we also want to have this answer because it relates to the next question, which is like, if you culture the cells on MatrixPlus, can you cryopreserve them, and will they retain their mature phenotype? And those are two questions that we still need to answer. But you know, like sometimes people are already doing it, and if you already know this answer and want to share with us, we would be really happy about that. So I think that this is the end of my presentation. And if you have questions, I would be pleased to answer them.
Excellent. Thank you all for joining, and thank you, Todd and Andre, for speaking today. Even though I've get to talk to you guys all the time, I always enjoy hearing from you guys. With that, if there's any questions, you can type them into the chat, or you can just speak up. But if you type them into the chat, I'll read them out loud, and then we'll answer those for the group.
I also wanted to say a couple of things while we wait for some questions. Guys, Andre, Todd, and Travis, thank you so much for that information. I know there was a lot of information that we covered today, so we're going to have a copy of the recording available on our website. We'll also send it to you via the email that we have on file for you. And you can also find a copy of the plating protocol that Andre and Todd both referenced for Fujifilm. It's on the homepage. If you go down towards the middle, you'll see it on the protocol section. And then you can also find a copy of the publication that relates to this. If you just go into our peer-reviewed literature, you'll find that there. And as Andre mentioned, if there's a particular format that you use that you don't see on our website, you can chat with us on our webpage using the chat function, or you can email me like Andre mentioned, and we can look at and check feasibility for you to see if we can coat those plates for you. It usually takes about 6 to 8 weeks for customization, but we definitely will want to help you with your experimental design if you get to that point. I see we might have a couple of questions, and I'll let Travis go ahead and take those.
Sure. So the first question is, why do we only sell Salvo MatrixPlus in plates and not in other presentations like Matrigel that comes in a liquid presentation? And so that's actually a really wonderful question that fortunately I have a good answer for. One thing that's really fascinating that we found in studying this is that actually the way these extracellular matrix proteins are presented to cells is actually critical to maintaining function, at least for certain applications. There may be applications where it's not necessary, but there's actually a paper coming out of our scientific founders lab soon, and it's not looking at the cardiomyocyte application, but what they've shown is that there's a protein in the bone marrow matrix that's critical for supporting osteogenesis. And if you downregulate that, you lose that ability, and adding it exogenously doesn't rescue the function. However, if you have disease cells that don't have that, and you can upregulate it using adenovirus, and when you upregulate it so it's integrated into the matrix, you restore the function. And so we can't make the assumption that we can solubilize these proteins and maintain their biologic function. And then also, again, it's so difficult to parse out mechanism of action when we're talking about complex matrices that we really still don't know what contribution to this phenomenon is mechanical and what is structural and what's biochemical. And so we do our best to leave it all intact. And so that's really the explanation for why we sell it this way. So it's maintained exactly the way the cells secreted it, and proteins are presented as they are in the intact matrix.
If there aren't any more questions, I want to also I know we've already gone over the time a little bit, but I want to also remind everyone we have our third session coming back on November the 4th. It's going to be at 1 p.m. Central Standard Time. We will also have that as a recording available for you if you're not able to make that time. But if you registered for the webinar at all, you'll have those all in your calendar already. We thank you so much for having you guys here, and we appreciate all the questions. And if there are any other additional questions, feel free to reach for us directly. We are happy to continue the conversation. Thank you, everyone. I'll go ahead and end the webinar now. Thank you.
All right. Thank you. Bye.